Hello students, very good evening. So before we start the session, I will launch a technical poll. So please let me know if my audio and video is clear and even the visibility of the PPT is perfect. So let me launch a poll for you. One second. Yeah. So just take a minute to just give me confirmation if my audio is clear and is the blackboard visible clearly and including the video. Okay. All right. So let me check with the YouTube. Okay. Perfect. So let's start our session. All right. So today we are going to start a new topic that is microbes in human welfare. We'll talk a little bit about this topic and we'll solve some questions with respect to this. As usual, all the questions that we are going to solve is going to be previous year questions. All right. So let's start from here. So when we talk about microbes in human welfare, so here is the outline of the chapter. In today's session, we are going to be talking about microbes in household products, microbes in industrial products, and sewage treatment, and the further topics we'll be taking in the coming class. Okay. So when we talk about microorganisms, the discovery of the microorganisms itself is a very huge Pandora box, I can tell you. So before this era, hold on, just give me a second. Yeah, questions. We're going to solve questions. 1 second, because this is the fresh topic we're going to talk about. So we'll finish this topic for 20 minutes or 30 minutes. And rest 30 minutes, I have questions prepared for you. So we can answer those questions, which are previous year questions. So this is the first time I'm talking about the topic. So let's talk a little bit about the content and then we'll solve questions based on it. Is that fine? Yeah. All right. So 1 second. Yeah. So when we talk about microorganisms, so the discovery of microscopy led to a huge potential for us to understand what we're interacting with the things that we cannot see. Correct, right? So, but if you look at the human interaction with microorganisms, it did not start with the discovery of a microscope. From a very it's an age-old practice. It's come from traditions and customs. For hundreds and thousands of years, humans have been using these microorganisms for a regular basis, either on a daily basis or as a ritual basis or a tradition basis. We keep on using this. So what are the other things that we can look around, the good aspects of microorganisms when we think about them? So if you are consuming yogurt or curd or buttermilk on a regular basis or sometimes, so that is because of the microorganisms. The softness of the bread or the bun or all the bakery products that you use, all of them is because of microorganisms. The Swiss cheese or the Roquefort cheese that we use, okay, it is also from microorganisms. The infamous toddy, nira, okay, and the famous fermented Bengali fish, it's all from microorganisms. And the everyday breakfast, go-to breakfast like idli and dosa, the softness in idli and the poor nature in the dosas is because of microorganisms. Okay, so these are some of the applications of microorganisms that we see on a daily basis, which has been used for several centuries right now. Okay, so let us look at other aspects of microorganisms, including these ones, and understand the benefits of microorganisms in a small gist. Okay, so here we go. Yeah. So one of the most common friend for us is lactic acid bacilli or lactobacilli. So lactobacilli, the very common application or help by this lactobacilli on a daily basis is conversion of milk into curd. So what it does is it produces these acids that coagulates and partially digest the milk proteins. So the partial digestion of milk proteins and the coagulation leads to the formation of curd. Okay. Whereas the same thing, the fresh milk is converted to curd, and through curd we extract butter and ghee. Correct, right? So it's all byproducts, the dairy byproducts. So the lab also helps in increasing the vitamin B12, which is very much essential for your bone strength. Okay, that is also produced by this lab bacteria. And the lactic acid bacteria or lactobacillus is also found in many of our parts of elementary canal, such as stomach, which helps in controlling the pathogens. So, the same lactobacilli is also found in female genital area, where it produces lactic acid. So, this lactic acid, what it does is it prevents any possible microbial infections in the urinogenital tract of females. Okay, so when we talk about the anaerobic bacteria, that is bacterial fermentation, the application of this anaerobic bacteria is also used to make foods like dosa or idli on a daily basis, right? So the puffed up appearance or the softness that is given by these bacteria is due to the liberation of carbon dioxide during this metabolic process. You know that in anaerobic reactions we have glycolysis. Okay, so here what happens, the fermentation takes place in the absence of oxygen, so that leads to the elimination of carbon dioxide. So the carbon dioxide production leads to the softness or crispness in the dosa. Okay, so next one is the Bekar's yeast. Okay, hold on. Which micronutrient is used for muscle building? So yeah, it's entirely different topic. You need proteins, which is very best to build your muscles. Okay, so you are asking micronutrients or micromolecules. So proteins are best, which includes most of the amino acids, because majority of your muscle building happens because of um, proteins. So that's the proteins are best. So some of the proteins are amino acids like branch chain amino acid or BCAA. Okay. Or you can just go for whey proteins. So they can help you in building muscles. Natural foods also, like for example, if you want the vegetarian users, you can go for mushrooms because they're very rich in proteins. And if you are going for non-veg, many meat contains proteins as their main nutrient. Okay. All right. Is it okay? Yes? No? Yeah. All right. So basically, we don't have microbes helping us. The microbes do have proteins we call SCPs, single cell proteins. It's just quite expensive though, but they also help in increasing our body or muscle mass. Okay. Next one is Bekar's cyst, for example. Are we in the line? Let me check. Yeah. So another example of anaerobic respiration that we use on a daily basis is yeast. So yeast is used in many of the bakery products such as bread, bun, or any of these puffed or cushy things that you eat. Okay, it's all because of this fermentation by yeast. And the yeast name, the common yeast that we use is Saccharomyces cerevisiae, which is also called as Bekar's yeast. Okay, so that also produces carbon dioxide, which gives that fluffy nature to these bakery products. And the famous toddy, which is used in many of the South Indian traditions, okay, in several states such as Andhra Pradesh, Karnataka, as well as Tamil Nadu. Okay, that is also produced by fermentation of sap. Okay, so palm trees usually to see palm trees, probably people from Kerala and people from some parts of Andhra Pradesh, they can find it very easily. So these palm trees, what they do is they take the parts of the palm and then they ferment it, the sap of the palm. So that is when, give me a second, students, excuse me. Okay, no problem. Okay, so now when the sap is fermented, you get toddy. Okay, next one. The in Bengal there is also fish. The fish is fermented and is being used. And then you have fermentation of soybean, fermentation of bamboo shoots, fermentation of rice, okay, or paddy, and then fermentation and also to production of cheese. So Many of these anaerobic applications are very famous, okay, in several aspects. So that is where the traditional use of microorganisms can be clearly seen, like toddy or fish or soybean or bamboo seeds or even rice. Okay. Now the cheese, which is very, very famous, and it is a commodity of luxury for us. Whereas if you see European countries, they really make good cheese. And one of the famous cheese that you have seen in cartoons, like Tom and Jerry, it's called Swiss cheese with lots of holes. So the holes in the Swiss cheese is also because of the carbon dioxide production during fermentation by a specific bacterium, and the bacterium name is Propionibacterium shermani. So this Propionibacterium shermani is the one that is used for the production of Swiss cheese. Okay, and another variety of cheese is there that is called as Roquefort cheese, which is one of the expensive cheese. Okay, so it is manufactured by ripening or the cheese is produced by ripening of a fungus called as Penicillium roqueforti, okay, on them. So while we do this, the Roquefort cheese is produced. So remember the organisms like Swiss cheese for Propionibacterium shermani and Roquefort cheese Penicillium roqueforti. Okay, next one. So these are the household products. So in household products, we understood. So it's very, very common because we will be able to answer this easily because we have been using for a very long time, such as lactobacilli for milk to curd, okay, and vitamin B12. And also, it also helps in checking the pathogens in stomach as well as female genital tract. Next one, the microorganism is also used for fermentation, production of dosa or idli, or even the bakery materials or bakery food such as bread by using baker's yeast. And the traditional applications of this by fermentation is toddy or fish, soybean or bamboo shoots. Okay, and also for the manufacturing of cheese. So these are all household applications of microorganisms. Now you can see that can you really date back when the humans learned to convert milk to curd, okay, and they really understood that I can use this lactobacilli to my advantage to make the best curd, not this completely spoiled milk, right? So that knowledge was discovered long, long ago before we understood that microbes were around us. Okay, next one, application of these microorganisms in industries. So what do we have in industries? For example, production of beverages, okay, so which is quite profitable for governments as well as people. Okay, production of beverages, antibiotics, okay, on industrial scale. So they are produced. For example, if I talk about industrial scale, just take an example of beverages. Look at the volume of beverages that are being produced in our own country. It's tremendous, isn't it? So the consumption is also high and the production is also very, very high than we can imagine. So that is why I say that for growing microorganisms to that extent, you need a specialized industrial setup that includes fermenters. What are fermenters? Fermenters are large vessels where they control a specific climate and aseptic conditions for microbial growth. So these fermenters are so large. So what they do is they maintain pH, temperature, okay, aerobic or anaerobic conditions, okay, and also they remove toxins and they make sure that there is a steady supply of nutrients for this microbial growth. So at this point, what will happen? The fermentation will take place without any sepsis or without any contamination. Understood. So take for example some of the industrial products that are produced. So breweries, brewery or brewers yeast or brewing things is very very recreational in many societies and traditions. So basically, what happens here? The beverages are produced by fermentation. For example, if you take beer, it is done by barley, and then you have wine that is grape, and some uh, industries use rice also. So basically what we do is we either collect cereals like barley or we go for fruit juices like grape is the very commonly chosen, or apple. Okay, so they are fermented to produce ethanol. Okay, ethanol is consumed like alcohol. Okay, example is wine and beer. Okay, wine and beer. So you just take grapefruits or you just take barley and you ferment. What do you get? You get the fermented grape juice and you get the fermented barley. So here what happens here, there is no distillation. That means you don't concentrate this alcohol using another industrial setup. So that is why when the wine smells like grapish and the beer smells like barleyish. Okay. So that but if you see the other distillated drinks like vodka or rum or whiskey, so all of them are distilled. Okay, it means they're concentrated from their broth. Understood? So these another brewery examples, brews example is whiskey, brandy, rum, gin. Okay, so attack. Uh, they are the different types of distillated drinks. Okay, which they are concentrated from the fermented broth. Each of them have their own techniques and each of them have their own environmental setup for the production. So you can see the amount of the industry that has been developed with respect to breweries by the application of microorganisms, right? So next one is antibiotics. So we know that antibiotics are sourced from another microorganisms. It is not by accident that magically we get antibiotics. So we have to isolate them. What are antibiotics in our nature? We know that there is like how microorganisms can infect us and cause a disease and make us weak. Similarly, for other organisms also they can cause disease, right? For example, if fungi is like us and microorganisms attack fungi and basically try to kill it. Now, how can fungi keep quiet? It doesn't keep quiet. So what it does is it develops its own toxins. So these toxins will kill this bacteria. So for if we take the same toxins to try to kill that particular bacteria, it will work for us. Okay, but that is a famous experiment and the famous story that we have with respect to microbiology. Correct, right? So antibiotics are basically chemical substances that are produced by microorganisms that can control or kill the growth of other microorganisms or pathogens. Okay, so we use several antibiotics. One of the common antibiotic that we use is tetanus, anti-tetanus injection, and other antibiotics like plague antibiotic, whooping cough, diphtheria, and then you have leprosy. So all of them are are antibiotics which control the growth or proliferation of these microorganisms. So we, as I told you, we have the famous story with respect to antibiotics. Alexander Fleming, okay, you know that he was working in his laboratory and he had plates of this, streptococcus. And suddenly he just saw one day that the streptococcus colonies disappeared like this. Okay, this white portion is streptococcus. Okay, so you can see that the colony actually, the penicillium colony, when it grows, the streptococcus was completely disappeared. So when he found this, he did not throw the plates. He did not think that, okay, there is a contamination, I need to throw it. He just saw that wherever this penicillium grow, the streptococcus bacteria did not grow. Penicillium is a fungi. Streptococcus is a bacteria. So why it did not grow together? Because whatever the streptococcus is, I mean, like the penicillium is producing, the streptococcus is very sensitive to it, or it is dying. So that is why it is not growing. So that led to this revolutionary idea of discovery of antibiotics. Okay, so streptococcus he grew, and the organism that inhibited or stopped the growth of streptococcus was penicillium notatum. So finally what happens is Ernst Chain, Howard Florey, okay, so they actually isolated the first penicillium. Alexander Fleming was given the credit for discovering that anomaly, and these two isolated antibiotics. And finally, all three were given Nobel Prize in the year 1945 for the discovery of antibiotics. Understood? So antibiotics are very, very useful. Actually, we are running out of antibiotics, but still, it has saved several millions of lives across the globe. Okay, during world wars, during whenever we have natural calamities. Okay, so a tetanus infection is not so easy, and many lives have been saved because of the discovery of this tetanus vaccine. Okay, so just give me a second. Okay, fine. So now let's go back to the application of microorganisms in industrial products. Okay, so when we talk about application of microorganisms in industrial products, first one we have is organic acids. So when we talk about organic acids, just remember, okay, we in left we have the microorganisms, and in right we have the organic acid that we isolate from them. So Aspergillus niger, a fungi, is used for the production of citric acid. Acidobacter aceti, acetic acid. The name itself says Clostridium botulicum, butyric acid. Lactobacillus, lactic acid. Okay, so don't get confused with Aspergillus niger to lactic acid or citric acid. Okay, so it's a common mistake you guys make. So remember, either of the columns that you are closed. So just go with the flow. Aspergillus niger, citric acid. Okay. Then you have Acetobacter aceti, acetic acid. Clostridium botulicum, it is butyric acid. Then you have Lactobacilli, lactic acid. So how do they produce? See, whenever we have an organism and they have a metabolism to perform. So when they perform their metabolic pathways, the byproducts of this metabolism would they produce like citric acid, acetic acid, or lactic acid. So there also will be conditions that can be provided for the specific organic acid production to improve. For example, if at this temperature, at this food source, at this cultural conditions, if Aspergillus niger is producing citric acid, what will you do? You will provide the same such conditions for this Aspergillus niger that promotes the growth or that promotes the production of this organic acids. Correct, right? So that is why we mimic the conditions for this microbial growth to give us that particular organic acid that it is capable of under certain conditions. Okay, so these are the examples. Next one, uh, the you know microorganisms in alcohol production. So in alcohol, what we use for wine, for example, we use yeast. Okay, and so Cerevisiae. S. cerevisiae is used for the production of Saccharomyces cerevisiae. Saccharomyces cerevisiae is used for the production of ethanol. Now, this is very important. Even though this is easy, like you have grapes. Grapes is fermented by Saccharomyces cerevisiae. Saccharomyces cerevisiae, okay, to produce ethanol. So it's very common example that we can remember. Next one is enzymes. So here is where a little bit of importance come. So some of the enzymes are also produced using microorganisms such as detergents. So they are used by they are produced by certain formulations. So they are used for removal of oils and laundry. Next one is pectinases or proteases. For example, you have Minute Maid juice, orange juice, right? Or Fanta. Fanta they don't use, like Minute Maid or any of the fruit juice like Tropicanas that you drink. You don't get the fruit pulp, right? You get the clean juice. But if you make the same juice in your home, you don't get that. You have a lot of colloidal kind of things floating around. So that is because we use proteases and then pectinases, which digest all this pulp and plant, fruit products and leave you the clear juice. Understood. So that is for clarifying bottles of juice, bottle juices. Next one is streptokinase. Remember this streptokinase. This streptokinase is used, for removal of blood clots. Okay, for clots. So it is isolated from streptococcus. Okay, streptokinase is produced by streptococcus. And what do we use it as? If we use it as clot buster, okay, for removal of clots, especially people who have heart attacks. What happens? So these people will have clogged blood vessels, right? Especially coronary arteries. So to relieve these coronary arteries, what do we use? We use this streptokinase so that it relieves their blood flow. Okay, next one is cyclosporin. Cyclosporin A is basically an immunosuppressive. Immunosuppressive means it reduces your immune capacity. What is your immune capacity? For example, I take somebody's organ or a tissue or any graft as a transplantation, correct, right? When I perform the transplantation, what happens is, so that particular area might get infected, correct, right? That particular area might get infected. Now, why it got infected? Because your body is able to differentiate between what is self and non-self. Simple. So it is able to understand this is not the one that belongs to me. That capacity is reduced by cyclosporin. So the cyclosporin makes your immune system to become quite, defenseless. So that is called a suppressing immune system. So it is used as immunosuppressant agent, okay, during organ transplants or any of the transplantation. Okay, so the cyclosporin is isolated by trichoderma polysporum. It is a fungus. Okay. Trichoderma polysporum is a fungus. Next one is statins. Statins are produced by Monascus purpurus. It is a yeast. Okay. So the use of statins is it is a blood cholesterol lowering agent. So basically, it is responsible for inhibiting an enzyme that is responsible for production of or synthesis of cholesterol. So if this enzyme is gone, then the cholesterol production won't take place. So that is why the statins are very, very helpful. Understood? So microbes and seepage treatment will take it in the next class. So till now, do you have any doubts? Can we go for questions? Yes? No doubts. Good. Thank you. Sandeep, where are you from? Which school? Sandeep, right? Vishakhapatnam. Okay. Where are your friends? Okay. Others, I know. All right. So let me open another PPT. One second, students. Just yeah. One second. Yeah, let me see. I have only joined the last classes. Okay, only you are joining for even for the past classes. Oh, it's only you. All right. From it used to be class 11. Yes, yes. Okay, okay. I get it. I get it. That's what I was seeing. Like few days, Vishakhapatnam was missing. So it's you then. All right. Good to know. Okay. Can we start our question rounds? All right. So yeah. Sorry. So here is our first question. Among the following pairs of microbes, which pair has both microbes that can be used as biofertilizers? Aspergillus and Rhizopus, Rhizobium and Rhizopus, Cyanobacterium and Rhizobium, and then you have Aspergillus and Cyanobacterium. So I will launch you MCQ poll. Okay, so just give me 1 minute. Let me launch an MCQ. Yes, the MCQ poll should be live. Okay, I hope you can see it. If you cannot, let me know. Yeah, you can see it then. Yes, that is absolutely right. Okay, your answer is answer C. That is Cyanobacterium and Rhizobium. You're right. Okay, so that is the right option. So why it is right option? Like which among the pair of microorganisms can be used as biofertilizers? Cyanobacteria will fixes atmospheric nitrogen, and Rhizobium will produce, it is also responsible for atmospheric nitrogen fixation. Whereas Rhizopus is not belonging to this category, and also Aspergillus, which is a mushroom. Okay, so that is why these options are not correct. Just give me 1 second. Like 1 second, students. Okay. Yeah. So we have the another question. Okay, just go through this. Yeah. Which of the following statements about methanogens is not correct? They can be used to produce biogas. They are found in the rumen of cattle and their excreta. They grow they grow aerobically and break down cellulose-rich food. They produce methane gas. I know that was very slow in reading, but anyways, you got the right answer. Answer is C. That's what you were telling. Which of the following statements about methanogens is not correct? So your option that you have chosen is C. That is, they grow aerobically and break down cellulose-rich food. Yeah, methanogens are anaerobes. So that is why they are able to live in the rumen of cattle. Okay. And what happens is they produce methane gas as a byproduct along with carbon dioxide. Okay. So methane is a fuel. So that's why it is biogas. Okay. It can be used for cooking or for any domestic purpose. Okay. Among the following pairs of microbes, okay, this is done. 1 second, students. Let me. Okay, this has happened. I'm sorry about it. Just let me. Okay. Identify the microorganisms which is responsible uh, for the production of immunosuppressive molecule, that is a cyclosporin A. And you have options. A streptococcus, this is actually Streptococcus cerevisiae, this is the option they are given. Trichoderma polysporum, Clostridium botulicum, Aspergillus niger. Okay, you have your answer. Answer is two. Yes. So that is immunosuppressive molecule cyclosporin is isolated by Trichoderma polysporum. Can you tell me, apart from the first option, what about the Clostridium botulicum? It is used for the manufacture of. It is used for the manufacture of butyric acid. Very nice. Aspergillus niger, I will tell it is citric acid. Correct? I will share the border. Next one. Chalo. We have to match the following from 1 to 2. So we have list 1. List 1 says Aspergillus niger, Acetobacter aceti, Clostridium botulicum, Lactobacilli. And then we have the list 2, that is acetic acid, lactic acid, citric acid, and butyric acid. Okay. I doubt that you're reading the question. Okay. So anyways, I have to solve this. So the question is asking that we have to match things. So Aspergillus niger is used for the production of citric acid. So I'll write it as 1. Acetobacter aceti is acetic acid. So I'll write it as 2. Clostridium botulicum is for butyric acid. So I'll write it here as 3. And lactobacillus is lactic acid. So that is 4. Okay. So if you check this, it is, what do we have? Option 3. Okay, so A is like this. Okay, so if you check like this, you will get the right option. So I think this Vishakhapatnam student, you are in cheat code. Okay, yeah. Which of the following is put into an anaerobic sludge digester for further sewage treatment? Okay, floating debris, effluents of primary treatment, activated sludge, and primary sludge. Okay, not on cheat code. Okay, we have the next question anyways. Which of the following is put into anaerobic sludge digester for further sewage treatment? Okay. Okay, activated sludge. So you have chosen your option as activated sludge. See, what happens during the treatment of water after the primary treatment? What do we have? We have secondary treatment. So here, what we do is we digest of all the organic waste that is left out in the water, which the job is efficiently done by microorganisms. Okay. So for that, what we have to do, see, let's say that we have tank 1 and we have tank 2 in simple terms. And these both tanks are closed tanks. So what is happening here? This is old water. This is new water. Okay. So this old water means already it's been under treatment. So what its treatment is almost done. So once the treatment is almost done, who does the treatment? Microorganisms, correct, right? Microbes does this treatment. So these microbes, what they will do is uh, it accumulates as flocks. So for example, bacteria, okay, bacteria or protozoa, okay, protozoa. So they form these flocks. So these flocks we use as activated sludge because they are very rich in the microorganisms that we need for, filtering out or cleaning this new contaminated water that is coming to the tank. So we need microbe growth, right? So we introduce this bacterial flock or the culture or the population into this fresh water that is coming. It's not fresh, it's contaminated, which is rich in organic waste. So we introduce them. So this is called as activated sludge. Okay, Vishakhapatnam, I told you're not cheating. You're on cheat code. Okay, so don't mind. So next one. Next question. For the commercial and industrial production of citric acid, which of the following microbes is used? So Aspergillus niger, Lactobacillus, okay, a Saccharomyces cerevisiae, Clostridium botulinum. Okay, so yes, perfect. So the chosen option is A, that is Aspergillus niger. Aspergillus niger is the organism that is used for the production of citric acid. So lactobacilli for lactic acid, Saccharomyces cerevisiae is for the production of ethanol, and then Clostridium botulinum is for bitric acid. Okay, okay. I think this we got it right. Okay, fine. Cyclosporin A used as an immunosuppressive agent is produced from Monascus purpurus, okay, Saccharomyces cerevisiae, uh, Penicillium notatum, and then Trichoderma polysporum. Okay, yes, we already answered this Trichoderma polysporum, which is a fungi, so that is the right option. Okay, which of the following can be used as biocontrol agent in the treatment of plant disease? Lactobacilli, Trichoderma, Chlorella, Anabana. Which of the following can be used as biocontrol agent in the treatment of plant disease? Do we have an answer? Okay, one second. Let me check your answer. Yes, perfect. Your answer is two, that is trichoderma. Yes, trichoderma is acting as a biocontrol agent. Why? Because trichoderma actually prevents the fungal growth. Okay, so the fungal spore development or the fungal infections that the plant suffers, trichoderma is very efficient in controlling its activity. Okay, so that is why trichoderma is correct. Select the correct group of biocontrol agents. Nostoc aspergillus nucleopolyhedrovirus. Next one, Bacillus thuringiensis, tobacco mosaic virus, and aphids. Trichoderma, baculovirus, and Bacillus thuringiensis. Oscillatoria, rhizobium, and then we have trichoderma. Which of the group includes biocontrol agents? You can answer early like before. I don't mind. Okay. Yes. Perfect. Three. Your option is three. So option three is absolutely right. Trichoderma, baculovirus, and Bacillus thuringiensis. See, if you see the question and if you see the chosen answer includes that something that helps plants. Here, along with something that helps with plants, some of them are there which can cause diseases also. Okay, so that is why oscillatoria. Okay, so that is why they do not include for what the question is asked. So trichoderma for fungi, baculovirus of okay, and Bacillus thuringiensis. You know, Bacillus thuringiensis we use for as cryogens or cryogens is used for the control of like bollworms, corn borers, okay, etc. So, which of the following commercial blood cholesterol lowering agent? Which of the following is a commercial blood cholesterol lowering agent? Our options are lipase, cyclosporin A, statins, and streptokinase. Yes, correct. So the commercial blood cholesterol lowering agent is called as statins. So statins actually prevent the enzyme that is responsible for formation of or synthesis of cholesterol. So that is why statins are used for the people who are having high blood cholesterol. Okay. A biocontrol agent to be a part of integral pest management should be. Okay. It's an interesting question. A biocontrol agent to be a part of integral pest management should be species specific and symbiotic, free living and broad spectrum, narrow spectrum and symbiotic, species specific and inactive on non target organisms. What do you have here? One. Chalo. You are saying species specific and symbiotic. Okay, species specific is right. How can you always look for symbiosis? Symbiosis is a very complex this thing, right? What do you tell? So wait, correct. What you've chosen right now is correct. So, anyways, I'll justify this. I'm halfway. So, symbiosis is a very complex, natural behavior of two organisms. It's a very complex relationship also. So, we can't expect that all biocontrol agents to be symbiotic. Even non-symbiotes can also become a very good biocontrol agent. Okay, free living and broad spectrum. So free living and broad spectrum. Broad spectrum is very nice. It is able to live on different organisms. And free living will not get our job done. Okay, so because it has to be depend upon our host. So when it is on our something, when it is dependent on something, then only it can act as a biocontrol agent. Narrow spectrum and symbiotic. Okay, again the symbiotic thing is not okay. Species specific and inactive on non-target organisms. This is a very ideal characteristics because one is it is species specific, and second one is whichever the organism apart from what we are trying to use them for, it is not affecting us. For example, we use Bacillus thuringiensis to avoid any of the worms. So now if humans consume or cattle consume, it does not affect these non-hosts, okay, or any other neighboring plants or something. So that is why species specific and inactive or non-target organisms is a very good characteristic. Next one. Which of the following statements about methanogens is not true? Okay. I think we had this in the beginning itself, right? So that's all. Actually, all the questions are, anyway, Sandeep, we just solved this in the beginning, right? Two questions, you remember? Okay. Yeah. So, Salam. So students, this is all the questions I could come across with respect to, whatever that is left or asked in microbes in human welfare. I don't have any much questions that I could come across. Okay. So we will stop